Dear students, moving on to the second part of biological control of insect pests. As the term indicates, which we have already discussed when in the previous lecture when we discussed about the introduction to biological control, that this is a method where living organisms are used to control the insect pests. The word itself, biological, means the living. Today I would be discussing the ways to increase the population of natural enemies, because when we talk about the biological control, these are actually the natural enemies of insects which are employed for the management of insects. Broadly, there are four ways to increase the populations of natural enemies. Number one, incorporation of natural enemies, that is termed as importation, augmentation, conservation, and application of microbial pesticides. See, biological control has its own advantages as it does not require further capital input, and being self-sustaining is cheaper than pesticides. However, it may be costly in the beginning as the natural enemies might have to be imported from other countries. On the contrary, other forms of biological control may be costly as every year capital has to be put in, so it is not permanent. Biological control has a scientific approach, has its long history since 1880s, and over the periods new skills and techniques are being added to maximize it. This control mechanism has now been fully conceptualized, that has taken long years of study and research. This approach requires detailed knowledge of the biology of pests and their natural enemies. Now, this is very important because until and unless we don't know the biology of the pest, their life cycle, their details pertaining to the different stages present in the development, and of course, their natural enemies. Because if we don't know which are their natural enemies, how are we going to use them? How are we going to employ them? This provides the practical manipulation in the field to keep the animal and plant growth under control. Third could be achieved through predation, parasitism, and diseases, etc. So, of the four ways, the number one method is the importation. Importation or classical biological control involves the introduction of a pest's natural enemies to a new locale where they do not occur naturally. So, in the importation, the predators or the parasites, whatever they are, the natural enemies are of that particular pest insect, are brought from outside that local area from where the pest is a insect is a pest. Early instances were often unofficial and not based on research, and some introduced species became serious pests themselves. There are examples where the species which was brought to the new area as a part of natural control itself became a serious pest. So this has to be taken care of. And how is it taken care of? It is taken care of by researching on it, by studying the insects in details, and then employing them as an agent of biological control. To be most effective at controlling a pest, a biological control agent requires a colonizing ability which allows it to keep pace with changes to the habitat in space and time. Control is greatest if the agent has temporal persistence so that it can maintain its population even in the temporary absence of the target species, and if it is an opportunistic forager, enabling it to rapidly exploit a pest population. One of the earliest successes was in controlling Eiseria purchasei, which is the cottony cushion scale, in Australia using a predatory insect, Rhodolia cardinalis, the Vedalia beetle. This success was repeated in California using the beetle and a parasitoid fly, Cryptochetum iseriae. Other successful cases include the control of Antonina graminis in Texas by Neodesmatia sangwani in the 1960s. Now, these are the classical examples wherein the insects, the biological control agents, were brought from outside the country and employed for the control of insect pests. Now, this is the beetle, the Rhodolia cardinalis, which is termed as the Vedalia beetle, was imported from Australia to California in the 19th century, controlling a very specific insect, that is the cottony cushion scale Iceria purchasei. Other successful cases include the control of Antonina graminis in Texas. I've just given this example. Damage from Hypera postica, the alfalfa weevil, a serious introduced pest of forage, was substantially reduced by the introduction of natural enemies. 20 years after their introduction, the population of weevils in the alfalfa area treated for alfalfa weevil in the northeastern United States remained 75% down. So it was able to control the pest uh, to a very great extent. The second method is augmentation. Augmentation involves the supplemental release of natural enemies that occur in a particular area, boosting the naturally occurring populations there. In inoculative release, small numbers of the control agents are released at intervals to allow them to reproduce in the hope of setting up a longer term control and thus keeping the pest down to a low level, constituting prevention rather than cure. In inundative release, in contrast, large numbers are released in the hope of rapidly reducing a damaging pest population, correcting a problem that has already arisen. Augmentation can be effective but is not guaranteed to work and depends on the precise details of the interactions between each pest and control agent. So augmentation is actually, it means the increase. So small numbers are released at intervals and then they are tried so that that they increase by themselves. They have an increase. They will increase in number and reduce the damaging pest population. An example of inoculative release occurs in the horticultural production of several crops in greenhouses. Periodic releases of the parasitoidal wasp Encarcia formosa are used to control greenhouse whitefly, while the predatory mite Phytocelius permissilis is used for control of the two-spotted spider mite. The egg parasite Trichogramma is frequently released inundatively to control harmful moths. Similarly, Bacillus thuringiensis and other microbial insecticides are used in large enough quantities for a rapid effect. Recommended release rates for trichogramma in vegetable or field crops range from 5,000 to 200,000 per acre per week according to the level of pest infestation. Similarly, nematodes that kill insects are released at rates of millions and even billions per acre for control of certain soil dwelling insect pests. So, different insects, for the control of different insects, the number which is used initially sometimes is low and then they go on increasing. This is what is augmentation. So of the four methods we have discussed the two methods, that is importation and augmentation, under biological control part 2. The rest we would discuss in the upcoming discussions. Thank you.